Okay, so this is the result from 1968 by by the student at Stanford at this time who wanted to be a chemical engineer and started writing these equations and realized that he really wanted to be a mathematician. So instead of doing chemical engineering, he became one of the nation's leading probabilists instead. Kurtz, Tom Kurtz, in Wisconsin. Okay, so let's just do the. Okay, perfect. So what I want to say now is there is also, and this is now I'm getting actually to the talk. You know, so I'm about to start the talk the way I want to. There is additional structure to this chemical kinetics or stochastic or non model. And this basically has to do with the linear algebraic structure that gets created by these equations. You can think about the numbers of species or molecules that are created and annihilated in each one of the reactions. And if you think about a vector of species, then these reactions are simply vectors of the appropriate net gains in your number of molecules. So this is called in chemistry, this is called stoichiometric matrix. And the stoichiometric matrix is a very important sort of structural equation that allows you to consider these biochemical reaction models. The talk that I am about to give is really about playing with these structural equations and trying to develop some statistical methods of inference on these structures. And the introduction that I just gave you is sort of an indication that at least in some circumstances we might be able to collect data that we'll get from these stochastic systems and then try to measure using these, so average data from many stochastic systems, we might want to measure how do these data fit within this stoichiometric structure. So in other words, you know, sometimes it is convenient to think about these equations as a realization of stochastic process. Sometimes it's convenient to think about it as differential equations. Sometimes it's convenient to think about it as vectors in the appropriate linear space. Okay? And all three of these representations are useful for various other purposes. So the type of problem that we've originally been interested in, and this is the problem that I started working on a couple of years ago. This was the sort of main project that I've been funded to do with the NSF, was as follows, that basically we had some number of measurements. Typically those would be measurements in the lab, typically those would be measurements based on some sort of RT-PCR, then it would be aggregated, but sometimes we would have the ability to actually dye some of the molecules that we're interested in and look at the fluorescent behavior of as a time series of certain biochemical species in single cell. These are very difficult experiments to do, but there is a couple data sets and there's a couple people who have done it. Most notably, there's a lab at Stanford and at University of San Diego of Smirny, who just written a beautiful paper in Nature in January about genetic oscillators. So the type of methods that they are using can be used to generate this kind of data. So basically, what is this? I have two types of molecules in my model. There's these T-prots and mRNAs. And I am assuming that I have the ability to observe the system at some fixed time point, okay? Either through fluorescence product or through some other products that I can basically use to defilter the noise and observe. I'm assuming that I'm observing the counts, which is obviously not true, nonetheless. So if I have this system now, if I sort of have an idea what the system could look like, I don't necessarily claim that I have all the equations. I maybe have some equations. And I have this data. Is it possible for me to try to say something about what is the most likely set of biochemical reactions that would fit this data? Okay, so based on the data, what is the most likely set of biochemical reactions that generated the data?
So chemical reactions are defined to the level of parameters? Or?
Yes, so the chemical reactions are defined in terms of these, okay, so let me go back, this is a good question. They're defined in terms of these vector, in terms of these vectors in the stoichiometric space. On top of this, obviously, you need the parameters that are these kinetic parameters. But say that I am not interested in kinetic parameters. Kinetic parameters can be computed in various circumstances in different ways. Some of these experiments might not be directly comparable, so if I compute kinetic parameters in one system, it doesn't necessarily mean that I will be able to repeat precisely the same type of experiment in another system and be able to compare the results. Typically not. So the more interesting thing for us was, well, is there a way to just make inferences about whether these parameters are zero or not? So in other words, significant, okay, as opposed to numerical value. So this is a sort of, okay, let me skip over this. So this is an example of a system which by no means is a chemical reaction system, but of which to some extent you can think this way if that helps you in achieving what you would like to achieve. So this is sort of a standard network that we have built using the Bayesian network inference method, and this basically looks at two different types of T cells activation patterns and two different types of mice. So I don't really want to go into this either. But the point is that this is created by means of some kind of Bayesian or likelihood method that allow you to create a graphical representation of interaction. Now you can think about these interactions in terms of chemical reaction model, if you want to. In other words, you can think about these interactions are basically being a way of decoding or coding these chemical reactions. You have a molecule, and that molecule gets consumed, creates another molecule, and so on. These graphs are very structured. They're very hierarchical because they are supposed to be representing certain biological process, meaning that you will at least expect that these subgraphs of these types, they will be very hierarchical. In other words, they will have at least partial order, which is reasonably well preserved in some branches of the. If that's the case, then the question very often that we're interested in is, well, what are the weights, or is there any kind of probability measure that I could put on these arrows here that would allow me to infer whether or not these particular interactions are real, or whether they're important, or whether they happen often. Okay. So this is sort of a way of explaining this in terms of biochemical reaction models. You can write out a biochemical reaction that will express that exact connections in that network. In this particular case, this is actually really simple because it's a linear network. Most of these interactions in both of these networks are linear interactions. So, meaning that they are describable by a system of ordinary differential equations which are linear. So, there's all sorts of things that can be simplified in our general approach. Nonetheless, what I would like to know now, I would like to know if I can put some kind of probability measures on these interactions, meaning if I can put some kind of probability on whether or not the particular coefficient in the reaction is zero. Okay? Because the coefficient of the reaction being zero basically means that the reaction does not proceed.
So practically you said that it could be described by a linear.
They absolutely could be. The only reason that they would not be is if you would have two or three different species giving rise to one, and then the way that this Bayesian network. The way that we train these Bayesian networks, we explicitly, try to discourage them not to do. So in other words, they were told to build these hierarchies. So from the chemical point of view, this is a linear network. It's a very complicated network, but it's nonetheless. Okay, so.